came and talked to me about joining the lab. She was coming with great credentials. So she had done her undergrad at the University of Michigan. She had already done a master's degree in Germany with Sonia Yannick, and just kind of really knew what she was doing. She also had really good mathematics skills. And so I thought, aha, I could get Angelika going on one of the most difficult, but potentially most important problems that we were facing at the time. So the lab, we had built these models that look mathematically at how different communities, organisms respond to floods and other kinds of disturbance. We had done this with plants and riparian vegetation. We've done it with fish. We really needed to do it with aquatic insects, but that was scary to me because I knew how many different types of aquatic insects are out on the landscape and how diverse they are. And so I had avoided doing that. Angelika came along, and I think she's really cracked the code. So this is an important problem because this information is needed to manage big river systems like the Grand Canyon in the south of the US. So aquatic invertebrates are the food base for the fish, for the birds, the bats, a lot of the ecosystem in that area. And until now, we didn't really have a tool to link flow management of this huge river to the production of different aquatic insects in the ecosystem. And along the way, as Angelika will show you, not only did she solve this and come up with a good modeling framework that we can now use, she discovered a few clever things that I think really we're only beginning to understand right now. One is there's a trade-off that's mediated by temperature between growth and development in insects. And utilizing that, she's able to go from numbers of insects to actually predicting the biomass of insects, which is hugely important. And a second thing, I'm not stealing all your thunder. Is something really she just discovered a few months ago, and that is there's a theoretical trade-off between the degree of competition within a species and among species. And by putting that little bit of information in her model, she's able to essentially see community dynamics within the context of the model that, in a way that I think we hadn't really thought of before. So it's both of those are really new areas of terrain that she's opened up that I really hope we expand on in the future. Unfortunately, Angelika's moving on. She has a postdoc at the University of Wisconsin, and she's going to start to fall. But hopefully we'll get a lot of papers out this summer. I have to wait on So with that, take it away, Angelika.
Okay, thanks, Dave, for that very flattering. Yeah, so I'm going to be discussing what I've been working on for the last 6 years, which is developing these aquatic invertebrate population models. And my clicker go. It won't. I do. How about there? Here we go. Minor technical difficulty. So I'm going to take a step back before we get into the weeds real quick to describe what I think is some of the most interesting and important parts of doing ecology and being an ecologist and how I got kind of involved in this in the first place. And that is describing natural phenomena. And there's a couple different ways that we can do this, right? So there's observational ways, which is usually where we start out. We see something interesting, we're curious about it. We can look at natural phenomena through an empirical lens, take it into the lab. And we can also describe natural phenomena using statistical or mathematical modeling frameworks in which we can basically simplify really complex systems into pretty nice numerical equations. And I'm interested in understanding how environmental characteristics impact organisms that live in specific areas and what those relationships look like. And I'm doing this through a framework of mechanistic models. And mechanistic just is jargon for basically saying we can use math to describe specific mechanisms behind patterns that we see in nature. And this means that we can take empirical and observational data and basically convert it into nice little pithy equations. And because I'm a freshwater ecologist at heart and I like playing in streams, rivers, and lakes, I'm looking at aquatic invertebrates. And aquatic invertebrates are ubiquitous, they're abundant, they're found in freshwater systems across the globe, they're incredibly diverse, and they're also really ecologically important. So they're the food base for pretty much the entire riparian system. All basically vertebrates in riparian systems, fish, reptiles, birds, bats, and predatory invertebrates as well, depend on aquatic invertebrates as their food base. And therefore, understanding what their population dynamics are going to be in different environmental conditions is really important for these systems. And I mentioned that aquatic invertebrates are diverse. And in fact, they're so diverse that often we categorize them into broad groupings via life history strategies. And this is basically the culmination of different traits. So these are traits like the fecundity, offspring per female, their lifespan, their susceptibility to different disturbances, physiological thresholds like temperature minima, temperature maxima. And we can use these to describe or basically broadly classify groups of organisms. So I want to you already just got a little introduction, but I want to give a brief outline of where the rest of this presentation is going to go. And I'm going to describe 3 objectives that align with the 3 chapters in my dissertation. So my first objective is to develop mechanistic models for different aquatic invertebrate life history strategies, looking at different environmental conditions. My second objective is to apply this to the Colorado River below Glen Canyon Dam, basically looking at how different environmental stressors impact populations within that ecosystem context. And my third objective is to then combine those single taxon models together into a large multi-taxon model and see how we can test different kind of theoretical biology assumptions about stabilizing, equalizing mechanisms within the system. And then I'm going to summarize like 6 years of work in like 2 slides. So that first objective is developing this mechanistic model for different aquatic invertebrates. And in order to do so, we have to ask some questions. So what parameters do we want to include in this model? And how do we want to construct this model? And because in biology and ecology, any question leads to more questions, that means we have to ask what drives invertebrate population dynamics? So we know invertebrates are ectotherms, they're cold-blooded, and so they're really dependent on temperature regime. Survival, growth rate, size of emergence, fecundity, lifespan, these all depend on temperature, and there's really strong temperature relationships. You can tell this here from some empirical work with temperature on the X, and we have survivorship, development time on the Y. There's pretty strong relationships being shown here between temperature and these vital rates. Additionally, we know that invertebrates operate under density dependence. And this basically just means that as the number of individuals in a population increases, the growth rate of that population decreases. And in kind of a mathematical context, we can describe that using carrying capacity, or K, which is the max number of individuals of a population that an area or a limiting resource can sustain. And then what we do is we can describe this using a time-varying matrix population model. And in this case, yes, I am showing that. Sorry, everyone. We have our matrix, AT, and we can multiply it by a vector of abundance, NT, and that steps us forward in time, giving us a vector of abundance at NT plus 1. And we can do that time step for time step for time step. And because we're working with invertebrates, we're going to use an invertebrate kind of scaled time step, which is 2 weeks. In this case, it's biologically relevant. Within our matrix, we have specific vital rates, and we describe them for different life history stages within our different invertebrates. So in this case, we have kind of like a larval, a pupal, and an adult stage. And if looking at a matrix of letters is kind of like gross, I can also show you a life cycle diagram, which might make this a little bit more intuitive, that as an organism moves through its life cycle, it can either remain within the same stage or transition into the next stage. And once it reaches adulthood, those adults make more stage 1s. And since this is time varying, we allow it to vary from time step to time step based on environmental conditions. So in this case, our stage 3 fertility it changes from time step to time step based on a density dependence assumption using a carrying capacity. And I'm going to flash some equations here, and if they're too painful, close your eyes. Just a warning. Additionally, we allow our probability of remaining in the stage or probability of transitioning to the next stage to be dependent on temperature. And so, yeah, that's what that looks like. And then we can relate abundance to standing biomass per time step. So the amount of biomass that's produced within the system within a 2-week time step via a per capita biomass, or basically individual body size. So we keep track of how long it takes an organism to go through its life cycle with specific temperature regimes. And so that allows us to estimate this per capita biomass. And then all we have to do is multiply per capita biomass times the abundance to get that standing biomass. And this gives us a model that takes a temperature time series and that matrix with vital rates. And you can play around with temperature, do whatever you want. And it gives us an output of abundance and standing biomass. And so for this first objective, we did it for 4 different life history strategies categorized by lifespan. So we have our boom strategy, which is typical of a beta mayfly. We call it boom just because they have boom bust dynamics in nature. We have a fast life cycle, a moderate life cycle, and a slow life cycle. And you can kind of see how we try to explore parameter space here. Degree days is a proxy for lifespan in insects. And then we can add a seasonal temperature regime. And in this case, we're doing a nice little temperate water temperature regime. So this is what you would find if you go out into a river or stream in Oregon. It's warm in the summer, it's cold in the winter. And we set the mean temperature to 14 degrees Celsius. This doesn't exactly matter for this one right here, but we're going to be shifting that mean in the future. And so you just want to know this is where we're starting, this 14 degrees Celsius. And then we run our model for 100 years, and we use the first 10 years as burden. And burden is just the modeling way to say we throw out that first basically 10 years of model output, because within those first kind of time steps, there's a lot of transient dynamics or complex dynamics that don't represent the long-term dynamics. And in the context of this project, we're interested in long-term dynamics. And so what we see is that when we add a temperature regime and we run them through the different life history strategy models, there's these large oscillations that occur. And we also see that there's seasonal cohorts that are being produced, right? So you can see here really clearly with the boom strategy, these distinct cohorts. And you might say like, wow, Angelika, that's like a lot. And I would say, have you been to the Midwest in the summertime? These are mayfly emergences occurring in the summer. This is a natural phenomenon that shows up on like Doppler radar. It gets kind of scary to drive. And so we recover these seasonal cohorts. And that brings us to wondering how these dynamics might change under different temperature regimes. So we look at cohort structures under different temperature regimes, looking at kind of a cooler temperature regime where that mean temperature is around 12 degrees Celsius, so a little bit cooler than this. And one where the mean is 20 degrees Celsius, so a little bit warmer than this. And then we can also move beyond cohort structure and look at mean abundance, mean per capita biomass, mean standing biomass, and basically look at how shifting the temperature regime up or down 10 degrees Celsius affects these kind of overall response variables. So looking at cohort structure, I just want to introduce this graph to you. There's 4 panels, one for each of the life history strategies. And we're looking at a year in the life of one of these organisms under either cool or warm temperature conditions. And as you can see right away, there's differences between the life history strategies. Kind of faster life history strategies have more cohorts. It's logical. Reproduce more within a year. But the big thing is that in warmer temperatures, more cohorts are produced. And this is something that occurs in real life. This is something that has been well documented in the literature. And it's something that I personally wasn't expecting to recover out of this model, but we did. Moving on to just shifting the temperature regime and looking at how abundance per capita biomass and standing biomass shift, we see that as temperature regime increases, the abundance increases. kind of up to the physiological threshold. So this point is basically when it's too warm and these organisms can't persist anymore. And this might kind of lead someone to believe, well, okay, so warmer temperatures, more abundance. Okay, that's not terrible. This happens in real life. This has been observed. However, on the flip side, per capita biomass actually decreases with increased temperature. This is also well documented in the empirical literature. And this means that our standing biomass, so actual biomass available for consumption in any given time step, is actually optimized or maximized at a lower temperature than our abundance. So this kind of restructures our thinking about what might be the most beneficial or most productive temperature regime in these systems. And this also happens in real life. It actually has a fun little name, the thermal equilibrium hypothesis. So to recap really quickly, this first objective, we developed these mechanistic models, and we can kind of reproduce fundamental aspects of invertebrate biology. We have these seasonal cohorts, we have shifting cohort dynamics. We can also link demography, abundance, to ecosystem level attributes like standing biomass. And we can kind of reproduce this trade-off between that abundance and standing biomass, this kind of like conflict between them at warmer temperatures. And this is relevant because freshwater systems are and will be impacted by differing temperature regimes in the future, primarily by climatic change. But there's also other stressors that impact these different freshwater ecosystems. And some of them are kind of more location specific than, say, global climate change. For example, damming. And so this brings us to the question of, can we apply this model beyond just kind of general temperature regimes to reflect specific taxa in specific environments? So my second objective was to take those mechanistic models and apply it to the Colorado River below Glen Canyon Dam. And I wanted to look at how different environmental stressors within this system impact populations of interest. And so now we get to reframe our questions to be, how does the Colorado River impact invertebrates? Also, what taxa should we include? So I want to give a brief introduction to probably one of my favorite places in the world, the Colorado River below Glen Canyon Dam. It's on this map right here. And this is a pretty large stretch of river from dam to dam. So from Glen Canyon Dam to Hoover Dam, it's about 305 river miles. And there's many stakeholders involved in working in the Colorado River, right? So this is part of the traditional homelands of the Havasupai, the Hopi, the Qualipai, the Paiute, the Zuni, the Navajo, or the Diné, and the Yavapai. And it's managed in cooperation with those tribes by the US government, agencies like USGS or US Geological Survey, Bureau of Reclamation, National Park Service, in addition to kind of energy stakeholders like Western Area Power Authority, and then obviously like state and local governing bodies. So there's a lot of people working in there. And so we know that climate change is going to cause pretty significant changes in precipitation patterns, in air temperature and water temperature, and a lot of other kind of ecosystem or climatic functions in this system. Additionally, the dam itself has fundamentally altered this segment of river. So above the dam is Lake Powell, which is a large reservoir. And so below, the kind of the discharge that comes out of the dam is artificially cold. It's pulling from the bottom of this deep reservoir. And there's really little flow variation compared to the natural monsoon and runoff driven flow regime. And in the past, there have been some high flow events for like beach building or sediment movement, and low flow events for like dam maintenance, but these don't really reflect any sort of ecologically relevant flows. And so we get to revisit what drives invertebrate population dynamics. So we know temperature regime, we know density dependence, and now we get to add dam-related disturbance. So the high flow events that they have, which happen kind of semi-regularly, basically look like this going along, and then they just kind of open the floodgates for like 2 days, and then they close them. And invertebrates have a really characteristic response to kind of flooding or high flow events. They're going to be going along in their life. Here's a high flow event. They have kind of major mortality. And then they see this resource pulse and they kind of just kind of like overshoot and have this huge kind of boom in population. And then over time, they recover back to like regular population. Additionally, you may have noticed those squiggles on that first hydrograph. This is because Glen Canyon Dam is a hydropower producing dam, and they match energy production with energy demand. And so on the daily, we have this increase and decrease in discharge to kind of match the energy use patterns of the people using energy from that dam. And this is called hydro peaking or load following. And this basically leads to this kind of like wave that propagates down the segment of the river, and it either inundates and drowns eggs that are laid on the river edge, or it exposes and dries out eggs laid on the river edge. And so oviposition location of a specific taxa is really critical here in kind of mediating risk and mortality related to hydropeaking. And so now we can kind of redefine our time varying matrix model by adding a flood-dependent carrying capacity into our fertility term, as well as adding any sort of kind of like mortality related to hydropeaking to relevant taxa. So we get to rewrite the math. We also allow organisms that are around at the time of a high flow event to experience some mortality. And then we just keep our temperature assumptions the same, right? So transition probabilities and probability of remaining are still temperature dependent in this system. So we identified 5 taxa that we thought were kind of ecologically relevant, really kind of big players in this ecosystem, and we parameterized them from the literature, and we did a lot of research looking at temperature-dependent growth rates, survival rates, high flow mortality curves, oviposition location, etc. Basically, we had to parameterize the entire models for them. And so we did that for some midge species of the family Chironomidae, for net spinning caddisflies of the genus Hydropsyche, for an amphipod Gammarus lacustris, for mayflies of the family Betae, and a fun fact, these guys, they're actually extirpated below Glen Canyon Dam, but they're found in segments directly above and also below that Glen Canyon Dam segment. So it's a fun little murder mystery there. And Potamomopyrgus antipodarum, the New Zealand mud snail, which is an introduced and clonal taxa in the system. And so this now gives us a time varying population matrix model where we can take temperature, we can take discharge, and we can take hydropeaking time series, as well as our vital rates. And we can get out abundance and standing biomass. This gives us basically 3 different levers to play around with. And we can then play around with them. And we sure did. So we came up with a bunch of different stressors to look at. So one of them is increasing temperature regime, right? So we have our baseline temperature regime, which is the average for the last 20 years. Then we have like about a 1 degree Celsius increase in water temperature. And this is projected under the representative concentration pathway 4.5, a climate change model. Then we have a plus 2 degree Celsius increase in water temperatures. This is projected under RCP 8.5. And then we have this kind of extreme 5 degree change in water temperature. And while it's not like necessarily projected under any given climate change model, it is technically possible because as the Lake Powell kind of decreases, we expect that water discharge through the dam will start being drawn from the top of the lake. If you've ever been swimming in a lake in summer, you know the top is warmer than the bottom. We also have seen kind of these extreme seasonal temperature swings. So in 2022, there was a temperature record that was 4 degrees Celsius above baseline. And for water temperature, that's actually quite large. So we created basically a spiked version of our 4 temperature regimes, where we just basically kind of added 4 degrees Celsius. Additionally, we were looking at high flow events, and this is actually what that looks like. And we modeled a 38,000 cubic feet per second, or about a 1,000 cubic meter per second high flow event, kind of in the mid-March, late March area every third year. And this is kind of on track with what historical kind of high flow event timing has been. And then we included either the presence or absence of hydropeaking at the same level of intensity as what occurs below Glen Canyon Dam. And so then we can just kind of play around with it. So we're only limited by time and computing power. It's a joke for the computer science. And so what we can do is look at kind of a multifactorial combination of stressors, and we can run it each of these scenarios through our 5 different models. We're doing the same 100 years, 10 years as burn-in. And then what we can do in addition is we can look at what stressor or combination of stressors has the largest impact on the dynamics of each of these organisms. And we do that by calculating an effect size, and we're using partial eta squared because this is common for multi-stressor scenario research. And so I want to present basically 2 results, at least with figures, the rest will be in the table. And I want to kind of introduce you to this image right here. So on our x-axis, we have temperature, kind of increasing from our baseline to our most extreme scenario. On the y-axis, we have scaled standing biomass. And so we scaled the mean to 0. So if you see negative numbers, there's not negative bugs. It's below the mean. And we do this because our organisms have different biomasses. So in order to compare them to one another, we want to make sure that our y-axes are standardized. And then we have our kind of rainbow of different scenarios that we ran through the model. And we also, whenever biomass goes to 0, we put a little asterisk just so we can tell that they basically kind of winked out in this scenario. And so what we see for our Hydropsyche species is that overall, as temperature regime increases, biomass decreases. And what we recover from the partial eta squared analysis is that the dominant stressor, actually the thing that had the highest partial eta squared, the highest effect size, is actually high flow event. And this is what divides these 4 lines from these 4 lines. And then a key interaction, so between multi-stressors, is this combination of temperature spike, and high flow event, which leads to this biomass dropping to 0 right here. And so we can do these for all the taxa. We see that biomass overall, as temperature increases, declines for our Betidae, our Chironomidae, and our gamma leucostrates. The dominant stressor for Betidae is hydropeaking. Their edge layers, thus they don't really do well under hydropeaking. Our Chironomids and our gamma leucostrates are highly temperature dependent. But our New Zealand mud snail does really well. So they actually see an increase in biomass as temperature increases. And this has to do with the physiology of this organism, which from empirical work doesn't really see any changes in growth rate, or at least negative changes in growth rate or survival up until around 34 degrees Celsius, which is basically bath water temperature. So yes, technically, the partial eta squared showed that temperature was the most dominant effect, but it's a positive stressor in this case. So to recap, when we apply these models to the Colorado River below Glen Canyon Dam, we see that hydropeaking and high flow events and temperature, they all have impacts on the biomass, but what the dominant stressor is taxon specific. And I get to show my favorite ecology meme up here, that whenever we ask a question in ecology, oftentimes the answer is, well, it depends. It's context dependent. It depends on the taxon that we look at. Additionally, this has kind of larger implications for the ecosystem that we're looking at, right? So we're speaking the language of communities when we look at standing biomass. This is directly related to energy flows available to those higher trophic levels, to those organisms higher up in the food web. And so if we start shunting biomass from our chironomids, which are really readily available for fish, for reptiles, for birds, for bats, to organisms that are basically only present in the water, that aren't bioenergetically available to non-aquatic species, so for our birds and our bats, we expect to see kind of changes. And this has pretty big implications. Because populations don't exist in isolation, they interact with one another. And so the taxa that we chose, they all occupy approximately the same place in the food web, the same trophic level. And it looks like it's a theoretical biology lecture. So within that same trophic level, taxa might compete for the same resource. And so they can do this in basically two different ways, right? So they're going to be competing with one another. These are New Zealand mud snails, actually, through intra-specific density dependence, where they're competing with individuals of the same taxa. Or they're going to be competing against individuals of a different taxa. That's interspecific density dependence. And about 70 years of theoretical biology has shown us that if a taxa or two taxa share the same resource, competitive exclusion occurs. And this is just a way to say one taxa wins. So even back in the 40s, if you put 3 beetles in basically like an enclosure and they all use the same resource, only one beetle taxa can win. Can kind of reach dominance and kind of competitively exclude the other two. However, this kind of leads to an issue because when you go out to a river and you look in a river, there are actually many different organisms that operate within the same trophic level that are there. There's lots of biodiversity. And this leads to the question, why is there coexistence then if competitive exclusion is supposed to be happening? And Peter Chesson suggested basically 2 main ways that this can happen. So the first way that this can operate is via stabilizing mechanisms. And this basically can be conceptualized using these resource curves. So I'm going to introduce these to you. On our x-axis, we have basically a resource gradient or resource axis. And on our y-axis, we have kind of the rate of resource consumption or the rate of use. And so if 2 taxa use the exact same resource, competitive exclusion is going to occur. However, if there's a way to pull those resource curves apart, there's a way to kind of differentiate, to partition those resources, coexistence can occur. And if you look at the mechanism behind it, what basically ends up happening is, in this case, intraspecific competition and interspecific competition are basically set equal. However, in a condition like this, intraspecific competition is actually higher or stronger than interspecific density dependence. And this is what Chesson calls a stabilizing mechanism. Conditions or situations in which intraspecific density dependence is larger or stronger than interspecific density dependence. The other mechanism that can occur is something that Chesson called an equalizing mechanism. So this is a situation where the growth and survival rates are basically kind of brought together for different organisms. And in a variable environment, or in a place with variable environments, we can kind of think about how there might be some conditions that suits one taxa better and other conditions that suit a different taxa better. And when there's kind of these differences in the environment, some will be doing well and some will be on the decline. And overall, long term, you can have coexistence of both taxa. And so this brings us to my third objective, where we want to combine those single taxa models that we made in the Colorado River into one large kind of community model or a multi-taxa model, and look at how different assumptions about stabilizing or equalizing mechanisms change the dynamics. And so in order to do that, all we have to do is rewrite the map. So in our single taxa models, we assume that there's basically a carrying capacity for each individual taxa. They don't interact. But what if we let the taxa compete for kind of a section of a total community carrying capacity? And since we're kind of speaking the language of communities, of ecosystems, we're going to use biomass because that's basically the standardized kind of value between all of these taxa. So we can rewrite our fertility terms. And then if we want to add a stabilizing mechanism, because it's math and we can kind of do what we want, we can just add a modifying term that basically changes the strength of intraspecific density dependence relative to interspecific density dependence. And that's this Zi term right here. So we can just add that and then toggle that as we want. And then if we want to think about equalizing mechanisms, all we have to do is add those stressors and look at variable environments. So basically what we end up with is we can take our kind of stabilizing mechanisms, either the Z sub I, so that that strength of intra-specific density dependence is equal to the strength of inter-specific density dependence. Or we can increase it so that the strength of intra-specific density dependence is greater than the strength of inter-specific density dependence. And we can run these through our multi-stressor scenarios. And we do this like we did all the other model runs for 100 years using those first 10 years as burn-in. And because we're working with about 70 years of theoretical biology behind us, we have some expectations. So my first hypothesis or expectation is that if that strength of intraspecific density dependence is equal to the strength of interspecific density dependence, so basically if that Zi term equals 1, competitive exclusion is going to occur. If the stressors are equalizing mechanisms under those conditions, we would expect to see coexistence. So basically, if those stressors are kind of enough to produce variable conditions and induce coexistence, then they're strong enough to be considered equalizing mechanisms. And our third hypothesis is if we increase the strength of intraspecific density dependence so that it's greater than the strength of interspecific density dependence, then coexistence should occur. That's right here. And so what we see is that when we set our Zi terms equal to 1, so when intra and interspecific density dependence are equal to one another, when they're all sharing the same resource, there is one winner, and that is the New Zealand mudsnail. And this is not surprising based on the single species models. And so that supports that first hypothesis that when inter and intraspecific density dependence are equal, that we would see competitive exclusion. However, when intra and interspecific density dependence are equal to one another, we really don't see any differentiation between our different scenarios. And so in this case, each of these panels is a different scenario. And we see very little variation. And this suggests that our stressors aren't really sufficient or strong enough in the context of this ecosystem to produce coexistence. I'll say that you could argue that coexistence is possible under high flow event conditions, under baseline temperature conditions, because we do see a slight coexistence with hydropsyche and New Zealand mud snails. It's pretty uneven compared to other forms of coexistence that we will see that this model can produce. So kind of overall, these stressors aren't really sufficient to produce a coexistence. Only like maybe one scenario is good enough. And then when we increase our intraspecific density dependence such that it's greater than interspecific density dependence, so basically that Zi term is greater than 1, we see coexistence, right? So we're adding in a stabilizing mechanism. Boom, we have coexistence. And so in this case, we can see that all taxa are pretty much present. There isn't one that's just kind of at that 100% at that relative biomass the entire time. However, notice that the kind of the rank, the actual relative biomass is dependent on the stressor. So it's not that these stressors aren't important, they're just not strong enough to be equalizing mechanisms. They still mediate dynamics within the system. And so you can see this really easily with our beta species under temperature and temperature plus hydropeaking. They're present with temperature, but their physiological response to hydropeaking, which is that they can't persist, puts them basically at 0 for that condition. So there is kind of an impact of these stressors in the context of a stabilizing mechanism. And so this supports our third hypothesis that basically adding a stabilizing mechanism does induce coexistence. So our third objective was basically to combine these single taxa models into a multi-taxa model. And we found that stabilizing mechanisms, when they're added, they do allow for coexistence to occur in this model. But the environmental stressors that we added themselves are not sufficient equalizing mechanisms in the absence of a stabilizing mechanism. However, they mediate responses of the organisms when there is a stabilizing mechanism present. So to kind of briefly summarize what I've been discussing for like the last 30 minutes, for that first objective in that first chapter, I'm looking at developing mechanistic models for different aquatic invertebrates under different environmental conditions. What we find is that we can do this. This model can also incorporate environmental fluctuations, and it can link demography abundance to kind of ecosystem function or standing biomass. When we apply these models to the Colorado River below Glen Canyon Dam and look at how different environmental stressors impact taxa of interest, we see that this model can be parameterized for individual taxa, and that we can kind of explore these different environmental scenarios to see how populations might change. And when we take it from abundance to standing biomass, we can then also relate it to the greater ecosystem. And then when we combine these models together into a multi-taxa model and look at kind of these more theoretical questions about stabilizing and equalizing mechanisms, we see that we can place ecological concepts like coexistence and competitive exclusion into this context of applied research. So looking at specific taxa and looking at specific ecosystems. And so I want to kind of leave you with one of my favorite quotes that I found while working on this research by George Box, a statistician, that all models are wrong, but some are useful. And like I want to acknowledge that these models that I've created, they simplify a really beautiful and complex system, but they're also useful, or at least I hope I can convince you that they're useful, because they link demography to ecosystem function. They kind of stand in the crossroads of applied and theoretical ecology. And because this is basically the beginning, as Dave mentioned, they can be used as a starting point for future questions about these ecosystems and about aquatic invertebrates in changing environments. So I want to acknowledge my funding sources. Thank you to Oregon State for providing a variety of grants, as well as the opportunity to work as a teaching assistant. Thanks, Lori, Carmen, Jeremy, Mark, Leslie. I didn't realize how much I liked teaching until I was a GTA and did the whole GCUT program. And I find it really wonderful that I got to experience that. Thanks to the Bureau of Reclamation, USGS, and National Park Service for funding my work and allowing me to do research in what I will personally argue is one of the more beautiful places in the world. I want to thank my committee chaired by my advisor, Dave. Dave, thank you so much for challenging me and also keeping me optimistic. And I'm pretty sure the imposter syndrome was gonna like annihilate me. I really appreciate that. I remember a very distinct phone call in an airport where I'm like, I can't do this. It's gonna be fun. We're gonna do it. I really appreciate that. Thank you to my committee for sitting through this talk and sitting through the next 2 hours. I appreciate your feedback in advance. I want to thank the Lytle Lab members past and present. Thank you for being wonderful colleagues, for going and doing field work with me, for giving me the opportunity to do field work with you. You're wonderful colleagues and also good friends. And I also want to thank the Novak Lab and Novak Lab members or Novak Lab meeting attendees. Mark, who can't be here today, gave me a lot of feedback on my dissertation and on my research, and invited me to his lab meetings. Thank you so much. Both you and the Lytle Lab read through a lot of first drafts and watched a lot of first drafts of presentations, and I appreciate all the time you invested in me. I want to thank all my friends, new, old, near, far. I really appreciate all of you. I'm so happy to be here. I love you all. I want to thank my family, most of whom are far, for also giving me a place to call home around the world. I love you all as well. Special shout out to Alex. Thank you so much for listening to me complain about mathematical bugs, coding and legitimate bugs for literally 6 years. A lot of what we did looked like this. Me on my laptop. I appreciate everything and your patience and support. And then I want to thank my parents. Yeah. Thanks for supporting me and encouraging me in always being a cool way. Anyways, I'll open up.
Thanks for that, Angelika. But before we take questions, I want to amplify what Angelika said. Our partners at the United States Geological Survey, the National Park Service, Bureau of Reclamation, because none of this would have been possible without loads of support from them. So, any questions from the audience?
So at the start of your talk, which was overall amazing, great job, a lot of hard work into this. You had talked about how many stakeholders are involved in this section of the river, but also just invested in this kind of resource overall. Based off your findings, what kind of recommendation would you give to folks? Okay, so like we're thinking like one on that of like you kind of mentioned that this is like the opening of kind of a new area to explore. Where would you like to see it go?
Oh, that's a really great question. So I think like one of the first places that this can go is linking and more specifically to better, sorry. Taking this from just the kind of assemblage or the population to actually looking at the kind of at least like multi-trophic level aspect. So we keep our carrying capacity pretty much the same. We just assume that it like is only changed by flooding. However, this is a kind of there's a seasonal dynamic here in resource availability to the aquatic invertebrates. So we can take data from NPP or GPP, so like primary production in the system, and you could actually like run that through the model and have a time step changing carrying capacity based on the actual like algal and like primary productivity that the invertebrates are actually based on to even like make that seasonality, I think, even more like acute. And I think that would be a really cool way to go with it, to like actually like include 2 trophic levels instead of just assuming a single carrying capacity.
I have 2 questions. One, so in the multi-taxa model, you had the Z equal to 1 and then greater than 1, what would happen if it was less than 1? Would that be like inter-specific competition stronger than
Yeah.
intra-specific, right?
Yeah. And so what probably, I mean, competitive exclusion would still occur in that case.
So kind of just be similar to if it was equal to 1.
Mhm.
Does that happen in nature? Can it happen? Like other things can. Hey, you know, it's a model, we can do it.
And then my other question was, if you could apply this to any other river in the world and maybe go do field work there, where would you want to apply this to other than the Colorado River?
Other river. I think that actually, so. Other river. Okay, I have my answer. I really am interested in, so Mount St. Helens on the kind of like secessional slope has a series of like small headwater streams that are like these high mountain runoff in this like secessional gradient from the volcanic explosion in the 80s. I would apply it there. I think that'd be very interesting to see. Yeah.
Is there any hope against the New Zealand mud snail?
So they're kind of the big well, they're a clonal introduced species, so they're all pretty much clonally the exact same. They came from like they're parthenogenic, so they just produce live young that are exact clones of themselves. And they have really strong, like they're just like really physiologically well suited for this system. In New Zealand, right where they're native, they have parasites that kind of keep their population in check. But at least as far as I've been able to tell, and in the literature, it seems like there aren't any parasites that our New Zealand mud snails have that really keep them in check in any significant way. Which means that they can pretty much reach these really high abundances. So I showed that slide with all those snails. I mean, that's what it looks like when you walk around in the river. So there's like nothing really besides, I guess, just like biomass available to consume. There's not very much like limiting their growth. And so, yeah, I mean, there's not like a great solution, I guess, to that, right?
You heard it here first, we need more parasites in town.
Yes.
Yeah, so thanks, Angelika. Great talk, and also I love that answer. Of course, we need more person. So my question was about your temperature responses. So one way in which the Colorado River in that beach is messed up is that it's much too cold, right? And yet all your organisms except for mud snail didn't like increases in temperature. What's going on with that?
Yeah, so that's the just the response of biomass. So overall biomass typically decreases with increased temperature in this specific system. And that just has to do with the they're already at small or at kind of like large size. So okay, let me work this through. So at cooler temperatures, organisms are larger. And then as you increase temperature, the per capita biomass decreases. Therefore, you kind of expect to see a decrease in biomass overall. However, there's some kind of you can get kind of some like windfall from abundance. So if your abundance can grow faster or enough, you can kind of either keep afloat or keep growing. But in conditions that are extremely stressed, like with hydropeaking and with high flow events, you don't get that kind of abundance increase. And that's what's causing this kind of overall decrease in biomass. So they're being exposed to these continuous stressors like hydropeaking, like high flow events, which continue to keep their abundances low. And then as increasing the temperature decreases per capita biomass, you then see that it's just multiplicative, that decrease in staining biomass.
Do you think that would change when you consider primary producers, which will surely produce more when it's more?
Yeah, I would expect hopefully kind of more of an equalizing response. But we have to run that first.
Okay, one more round of applause for Angelika.